Hello, dear Pharma aspirant. Welcome to GDC. As you know, we have started a Tejas series. So, Tejas series basically a series of important questions for railway pharmacist exam. In the last week class, we basically talk about carbohydrate and its metabolism and some of the important function test for different organs. So, today we basically cover the protein and its metabolism, nucleic acid. Let's come to the first question. So, first question is protein word is derived from protein word is derived from what? Proteins, proteose, proteins, yeah, protease. So here the correct answer is option number B. Protein word is basically uh, derived from proteose. So if we'll see the word protein is derived from the word proteose and this meaning is of the first rank. This term was coined in the year 1830 by a Swedish scientist John Brezila. So you have to remember the name as well. So the word protein is derived from the derived from where the word proteus, and this basically coined by the scientist John Brezilus to reflect the importance of this group of molecule. A stretch of DNA called gene carry the information required to build a protein. So basically, our the if question asks which cellular organelle is responsible for the production of protein, you should remember ribosome. Ribosomes are responsible for the production of protein. Okay, coming to the next question. Which of the following is a nutritional deficiency disorder? Okay, your options are MOMS, marasmus, mycosis, and myelitis. So, which is basically occurs due to deficiency of nutrients. So these are the option here. The option B will be correct answer because you might have about Quasius marasmus. This is a nutritional deficiency disorder. You can see marasmus is literally what means to waste. Okay, it mainly occurs in which age group? This basically affect our basically children's. Marasmus is predominantly due to deficiency of calories. If your food is basically deficiency in calories, so this will leads to development of marasmus. This is usually observed in children going in watery gruels or cereals to supplement the mother's breast milk. So you can see this is a typical marasmus patients. Okay, coming to the next question, who was the first person to identify Quasilcore? Quasilcore. Okay, so your options are Dr. Adam Maxwell, Dr. Gerland Quasilcore, Dr. Chris Williams, or Dr. William Winterhoff. So here the right answer is Dr. Cecil Williams. So what is this question called? So Jamaica pediatrician Cecil Williams is introduced the term 1935, 2 years after she published a disease first formal description. Williams was first to conduct a research on Kwashiorkor and differentiate in the other dietary deficiency. Kwashiorkor is a type of malnutrition. This is basically a type of malnutrition characterized by severe protein deficiency. So remember, Kwashiorkor disease is which is characterized by uh, malnutrition. This is basically due to severe deficiency of protein. It causes fluid retention and swollen distance abdomen. You can see the abdomen is basically swollen. Okay, so this is due to what reason? Due to accumulation of fluid. Okay, kwashiorkor is most commonly affect the children, particularly in the developing country with high level of poverty and food insecurity. Okay, coming to the next question, determination of amino acid sequence. So this is very important. Several times this question was asked. Which of the following which is responsible for the determination of amino acid sequence? Your options are Sanger's reagents, Biot reagent, mean hydrogen reagent, yeah, millions reagent. Okay, for the detection of amino acid sequence, basically Sanger's reagent is used. So what is basically the Sanger's reagent is? Sanger's reagent is a sequence of amino acid forming a backbone of protein, and local location of disulfide bond in a protein is called as primary structure of the protein. So this Sanger's reagent basically used for the identification of primary structure of the protein. Primary structure of protein. Amino acids are joined covalently. They are joined covalently by peptide bond. Protein biosynthesis basically starts from the N terminal end. So where it basically starts? This typically starts from N terminal end. This is also important. Question can be directly asked. The protein biosynthesis starts from which terminal of amino acid? That is N terminal, not the C terminal. Okay. Coming to the next question, the bond that holds the tertiary structure. This is very important. Which structure is basically present in the tertiary structure of the protein? We have four major type of structure. One is primary structure, secondary structure, tertiary structure, and quaternary structure. So quaternary question is tertiary structure is associated with which type of bond? So here the right answer is all of the above. All type of bonds are present in the quaternary structure because this is a complex structure of the protein. If we'll talk, the primary structure is formed by peptide bond. Primary structure is formed by peptide bond between adjacent amino acids. Secondary structure of protein is stabilized by basically hydrogen bond and disulfide bond. They are along with the hydrophobic interactions. Okay. Tertiary structure is nothing but next higher level of organism protein level. However, quaternary structure contains all above the structures. Coming to the question number 6, which amino acid can be synthesized by human body and not essentially included in our diet? So if we'll talk about amino acids, so amino acids are three types, or basically classified into three types. One is essential amino acid, another is non-essential amino acid, third one is semi-essential amino acid. Essential amino acids are cannot synthesized in human body. It has to take from outer source. It cannot synthesized in human body. So it has to taken as a supplement from the outer sources. Non-essential amino acid means this can be synthesized in the body. No need to take. Essential is not synthesized. Non-essential is synthesized. So what is semi-essential? Semi-essential means some of the amino acid can be synthesized in children but cannot synthesize in adult. So we call it as semi-essential. Question is which amino acid can be synthesized by human body? Non-essential to include our diet. That is basically our non-essential amino acid. Okay. Your option B is correct answer. Let me summarize what is essential amino acid. Essential amino acid cannot be synthesized by body and need to supply it through the diet. Example is valine, isoleucine, histidine, methionine, arginine, leucine, lysine, threonine, tryptophan, phenylalanine. Non-essential amino acid can be synthesized in the body. No need to taken from the external sources. Example is cysteine, alanine, tyrosine, asparagine, proline, aspartic acid, serine, glycine, glutamine, and glutamic acid. Semi-essential means some are synthesized in the can be synthesized in adult, but not can be synthesized in the growing children. Example is our arginine and histidine. Okay. Well, coming to the next question, Sakaguchi reaction. So, what is the usage of Sakaguchi reaction? So, for arginine detection of arginine protein, linkage in albumin, melanin, yeah, cysteine. Here, remember Sakaguchi reaction is basically used for the detection of arginine in protein. So, this basically detect arginine. Okay, what it detects? It basically detects the arginine in protein. Coming to the next explanation, the Sakaguchi test is a chemical test which is used for the detect presence of arginine in the protein. What it basically detect? It detect the presence of arginine in the protein. The name after the Japanese food scientist and organic chemist Soyo Sakaguchi. Arginine contain guanidino group. Okay, that react with alpha naphthol and alkaline hypobromide to form a red color complex. Okay. So alpha naphthol this is the formula of alpha naphthol. Okay. Coming to the next question, collagen fibers are rich in all of the following, but have extensively low content of what? Collagen fiber. So collagen fiber is one of the important fiber which is responsible for the elasticity of our body. What is its role? Elasticity. Elasticity. So which have extremely low content, all of the following but having extremely low content. Your options are glycine, cysteine, proline, hydroxyproline. Here, the right answer is cysteine because cysteine is present in collagen in very, very minute quantity. Okay. Coming to the next explanation, the amino acid sequence in collagen is generally a tripeptide. Okay, the collagen amino sequence is tripeptide, that is glycine, uh, glycine XY, where X is a proline, Y is for HYP. Only glycine residue can be accommodated at very tight junction between individual chains. The proline and 4-HYP residue permit the sharp twisting of collagen helix. Cysteine can disrupt the collagen helix and present very rare in collagen fibers. So, why question can be asked in other way? Why the concentration of cysteine is very low in case of collagen fiber? The answer will be cysteine can disrupt. So cysteine, what it does? Cysteine basically disrupts the collagen helix. So it basically disrupt the collagen helix. That's why it is present very less amount. In human, all of the following amino acids are essential except all are essential amino acid except uh, valine, isoleucine, glycine, and phenylalanine. Okay, so your right answer is glycine. Glycine. So let me tell you, glycine is basically comes under a uh, non-essential amino acid. Okay, that is comes under non-essential amino acid. Okay, moving to the next question. The example of metalloprotein. So which of the following is a metalloprotein? Casein, ceruloplasmin, gelatin, salmine. Which of the following is a metalloprotein? So your right answer is ceruloplasmin. Ceruloplasmin is a metalloprotein. So what basically metalloprotein is? So the protein which having heavy metals. If a protein containing a heavy metal ion are bound directly to some of the side chain of the histidine, if question is telling that the protein which having a metal ion directly bounds to the some of the side chain like cysteine, or some other amino acid are basically called as metalloprotein. Two metalloprotein, transferrin and ceruloplasmin, occurs in the globulin fraction of the blood serum. They act as a carrier for iron, copper, respectively. They are carrier for iron and copper, respectively. Ceruloplasmin. Okay. So example of amino acid having ceruloplasmin comes under metalloprotein is histidine, cysteine, and ceruloplasmin. Okay. So coming to the next question. See, so GDC has launched a crash course for a railway pharmacist, which is basically consist of 200 live classes, 500 plus recorded video lectures, 400 plus online test series, 100 plus previous year MCQs, along with you will get e-notes. Okay, so along with that, free marathon, pharmacist marathon has started on every day at 9 p.m. Okay, the price for this course has been reduced, as basically the actual price is 4,000, but due to offer it reduced it to 2,999. Okay, you can purchase this course for the preparation of RRB pharmacist. So coming to the next question, which proteins are called transporter proteins? Which proteins are called transporter proteins? Your options are 𝘂𝙎𝘾𝙐, hemoglobin, keratin, and enzyme. Transport protein means these proteins are involved in transportation of some of the material in our body. So you know hemoglobin is a transport protein. Why? Hemoglobin basically carries your oxygen. Okay, but it basically carries what? It carries oxygen. Question can be asked, one hemoglobin molecule can carry how much oxygen? Remember, one hemoglobin molecule can carries four oxygen atoms. Okay, this is you have to remember. So hemoglobin is the right answer. So storage protein is 𝘂𝙎𝘾𝙾 and glutamine. 𝘂𝙎𝘾 and glutamine are storage protein. Transporter protein is hemoglobin and serum albumin. Carrier proteins and channel proteins are the two major class of membrane transport proteins. Carrier proteins are also called as carriers, permeases, or transporters, but it binds to the specific solute to be transported and undergo series of conformational changes to transport the bound solute across the membrane. Moving to the next question, globular protein shapes. Globular protein shape are oval shape, cube shape, needle shape या square shape. Globular protein. So globular protein basically are oval shape. Okay, this is also you have to remember. On the basis of shape, based on the molecular shape, proteins are classified into two class: globular protein and fibrous protein. They are basically globular proteins are spherical or ovals in shape. They are early soluble, easily soluble. Example is your AGP. So you can remember AGP, albumin, globulin, and protamine. Albumin, globulin, protamine. So protamine is basically present in where? So protamine sulfate is a protein which basically found in sperm of fish. Okay. So protamine sulfate having what type of activity? It act as an antidote for heparin poisoning. Okay, fibrous protein, though these molecules are elongated or needle shaped, their solubility is very minimum. Example is your collagenase, elastin, and keratins. Okay, coming to the next question, secondary structure of the protein. These are basically consisting of alpha helix and beta plated sheet. Okay, secondary structure is consisting of alpha helix and beta plated sheet. This question also asked several times in exam. Okay, so the secondary structure of the protein basically consisting of alpha helix and beta sheeted protein. The spatial arrangement of protein by twisting of the polypeptide chain. This is basically due to twisting of the polypeptide chain. The spatial arrangement of protein by twisting of the polypeptide chain consisting of alpha helix and beta plated sheet. So this is basically consist of alpha helix and beta plated sheet. So moving to the next question. So next question is telling about the spatial arrangement of this subunit is known as spatial. When we are talking about spatial arrangement, we call that comes under which type of protein structure. When we talk about spatial arrangement, so remember primary structure protein have four structure: primary, secondary, uh, tertiary and quaternary structure. When we talked about spatial arrangement, so the quaternary structure is basically called as the spatial arrangement of this subunit. So you will see the sum of the proteins are composed of two or more polypeptide chain. Okay, this spatial arrangement of this subunit basically known as quaternary. Spatial means they are arranged like this. Okay, this is one subunit, another subunit also arranged like this. Okay, this is basically called as spatial arrangement of protein. So the quaternary structure is basically stabilized by, as I discussed previously, hydrophobic bonds, hydrogen bonds, and ionic bond and van der Waals process. Example: the example is very important from exam point of view. Directly question is asked: which of the following is a quaternary structure of comes under quaternary structure protein? Your hemoglobin, creatine kinase, and lactate dehydrogenase. These are comes under your quaternary structure of the protein. Next is 2,4-dinitrofluorobenzene. So this is basically used in Sanger's method, Edmonds method, Lawrence method, or Byrod's method. 2,4-dinitrofluorobenzene. So this is basically used in Sanger's method. Okay, this is basically used in Sanger's method. The Frederick Sanger developed a fast method for the identification of N terminals. This Sanger's method is used for what? The identification of N terminal of amino acid of a protein. In this method, N terminal amino acid is labeled with a fluorodinitrobenzene. So this is basically labeled with fluorodinitrobenzene that forms a stable covalent bond with amino acid. The protein is hydrolyzed and the amino acid is labeled, is identified. Edmonds degradation involves sequential removal of amino terminal residue by reacting with the phenyl isothiocyanate. Okay, Lawrence method and Byrod's methods are for the protein estimation. This Lawrence method and Byrod's method, this is basically used for the estimation of or quantification of quantification of protein. Sanger's reagent for detection of protein sequence. Next question is which of the following is not a waste product of protein metabolism? Which of the following is not a waste product of protein metabolism. Options are ammonia, creatinine, uric acid, urea. Not question in question the option is there as not, which is not a waste product of protein. So your right answer is uric acid. Because uric acid is a metabolism of urea cycle. Okay, so urea is a end product of protein metabolism. That is basically amino acid. The nitrogen of amino acid converted to ammonia, toxic to the body, and it is converted to urea and detoxified. Okay, so you can see urea cycle, also known as Krebs-Hansen cycle. Carbon dioxide react with NH4 ion to form a carbamyl phosphatase in the presence of enzyme carbamyl phosphatase synthetase 1. Now this carbamyl phosphatase convert ornithine to citrulline, that's citrulline to arginosuccinate to arginine, and arginine to ornithine. So during conversion of arginine to ornithine, one urea molecule is eliminated. Okay. Next, coming to the next question, amino acids are the end product of digestion of protein, fat, vitamin, carbohydrate. So this is very simple question. Amino acids are the end product of protein metabolism. Okay. So proteins are the amino acids are the building block of protein. Similarly, deoxyribonucleic acids are building block of DNA. Ribonucleic acids are building block of RNA. Monosaccharides are building block of polysaccharides. Fatty acid and glycerols are building block of uh, lipid molecule. Okay, you have to remember. Coming to the next question, synthesis of urea takes place exclusively in which part? Urea synthesis basically takes place in kidney, liver, urinary bladder, or pancreas. Remember, urea synthesis basically takes place in your liver, because liver is the major site for the urea biosynthesis. So the urea is a end product of protein metabolism. So urea synthesis basically carried out in the liver and transported to kidney for the excretion. Remember, in liver it is synthesized and transported to kidney for excretion. Transported to kidney for excretion. Okay. This cycle has already discussed. Again, I am repeating. Urea cycle is also known as Krebs-Hansen cycle, where carbon dioxide react with NH4 molecule to form carbamyl phosphatase in the presence of enzyme carbamyl synthetase 1. Now, this carbamyl phosphate convert ornithine to citrulline in the presence of ornithine transcarbamylase. Now, citrulline to arginosuccinate in the presence of enzyme aspartate. In the presence of aspartate, enzyme is arginosuccinate synthetase. From arginosuccinate to arginine in the presence of enzyme arginosuccinase, and from the arginine to ornithine, during this one urea molecule will be eliminated. Okay. Next is albinism. This is very important question from exam point of view. Albinism is produced due to the defect in biosynthesis of which compound? Tyrosine, melanin, valine, and isoleucine. Albinism. So albinism is associated with melanin protein. So albinism, Greek albino means white, is a inborn error due to lack of synthesis of pigment melanin. So due to lack of synthesis of melanin pigment, so this albinism basically occurs. This is basically an autosomal recessive disorder. Okay. So there is a two type. One is sometime I have in our class I might tell about some is recessive, someone is dominant. When one gene is dominant over another gene, uh, due to that a disease occurs, we call it as a dominant disorder. When one gene is suppressed due to and due to that there is a occurrence of disorder, any type of disorder, that is called as recessive. Okay, this is a autosomal recessive disorder. Biochemical basis, the color of the skin and hair color, hair is controlled by large number of this melanin pigment. The most common cause of albinism defection is tyrosine, the enzyme most responsible for the biosynthesis of melanin. So remember, due to the deficiency of enzyme tyrosinase, due to tyrosinase deficiency, so occurrence of which takes place albinism, because deficiency or tyrosinase defect affect the melanin metabolism or melanin biosynthesis, and due to that the albinism basically occurs. So the most important function of melanin is protect from the body from the sun radiation. Lack of melanin albino makes them sensitive, makes them white. You might see never some of the patients around your society. They are having this type of these types of marks. So this is basically due to lack of melanin pigment. Increased susceptibility, and these are basically increased susceptible to skin cancer. Photophobia, intolerance to light associated with lack of pigmentation in the eyes. Okay, so you can see some of the patient around you, your society. Okay, question number 20. So the fragment of DNA attached to the RNA initiator component was discovered by. So basically they are talking about what they are talking about. They are basically talking about Okazaki fragment. So Okazaki fragment what? So the fragment of DNA that found on the lagging strand. There are two type of strand, leading strand and lagging strand. The DNA fragment, the fragment of DNA that found in the lagging strand of the DNA replicate the fork known as Okazaki fragment after the scientist Reiji Okazaki who were discovered in the 1960s. The RNA initiator to which the Okazaki fragment are attached is known as RNA primers. The initiation of DNA replication has to start with RNA because the DNA polymerase can start DNA replication on its own but can extend it to existing chain where RNA polymer can. So, GTC also announced a batch All India Pharmacist Crash Course for BR DSSB, ASI Railway aims. All the pharmacist courses are covered in this. Okay, so this batch has started from 18th August 2024. So this also in this course is also in offer. You can get it in 2,999. What are the key features that you are going to obtain? First thing is live classes. Definitely you will get live classes by the esteemed faculty member. Second is recorded video lectures, online test series, previous year MCQ, along with e-notes. Whatever you will, whatever PPTs you are presented in your live classes, that also you will get as a e-notes. Okay. Coming to the question number 21, the chemical name of 2-amino-6-oxypurine. 2-amino-6-oxypurine, which of the following is the chemical name? Remember, this is the chemical name of guanine. If I will talk, guanine has one amino group at the position number 2 and one oxo group at position number of 6. Okay, guanine is the one of the four nucleotide base. So what are the four nucleotides basically present in the DNA? If we'll talk about DNA, the nucleotides we found are A, T, G, C. That is adenine, thymine, guanine, and cytosine. If we'll talk about RNA, A, G, C, U. Thiamine is replaced by uracil. Okay. Histone. So what is histone? Histone is a protein, whether it is a acidic protein or whether it is a basic protein. So a histone is a acidic options are acidic chromosome of the protein. Remember, I'm giving you some of the hint. Histone is a first thing is it is a basic protein. It is found associated with associated with DNA. It is found associated with DNA. Okay, associated with DNA, and it is basically coiled around a DNA to give a structural framework. Okay, so histone are the basic protein. They are basically rich in lysine and arginine. Your histone proteins are basically rich in lysine and arginine. They are basically bind to the negatively charged DNA molecule by the ionic bond. They are five type, that is histone H1, H2A, H2B, H3, and H4. So histone helps the DNA holding as the DNA wound around the histone core. Bitter appearance of the chromosome due to this histone DNA pulse known as nucleosome, separated by a small stretch of DNA known as linker DNA. The bead of each molecule contains 8 histone molecules, 2 copies of each H2A, H2B, H3, and H4. Coming to the next question, for DNA dependent RNA synthesis, a RNA template and DNA template. a DNA dependent RNA synthesis, a RNA template and DNA primer are required, a DNA template and RNA primers are required, a DNA template and DNA primers are required, or only DNA template required. So here the right answer is only DNA template is required. The DNA dependent RNA polymerase can polymerize RNA using a DNA template, DNA template of de novo without using any primers. Okay, without using any primers, DNA polymerase on the other hand requires a primer in the form of RNA for pre-existing DNA molecules. Okay, what is the ratio of constant ATGC? Remember the according to Chargaff's rule. This is basically decided according to Chargaff's rule. So Chargaff's rule is A equal to G, according to Chargaff's rule, A equal to T, G equal to C. And another one is A plus T equal to G plus C. The molar concentration of purine base A and G is equal to the molar concentration of pyrimidine base T and C, because one each type makes a base pair. Because of base pairing rules, A equal to G and G equal C, the specific ATGC are varies. Okay, another question is asked about uh, how many double bond between A and T. There is double bond is present between G and C. Triple bond is present. You have to remember like this. Nonsense codon brings about amino acid activation, initiation of protein synthesis, termination of protein synthesis, elongation of peptide chain. So nonsense codon are also known as stop codons. Stop codons. That's why they terminate the protein synthesis. Okay. Next question, which of the following is not a component of polysome? Protein, DNA, mRNA, tRNA, which are not a component of polysome. The right answer is DNA. DNA is not a component of polysome. Okay, coming to the next question, Lesch-Nyhan syndrome is due to X-linked SGPRT deficiency, X-linked dominant APRTase deficiency, X-linked recessive APRT deficiency, X-linked recessive SGPRT deficiency. This is basically due to, remember, Lesch-Nyhan syndrome is associated with SGPRT. First thing is, you are remember this is associated with SGPRT. Whether it is dominant or recessive, so it is associated with recessive SGPRT deficiency. Okay, Lesch-Nyhan syndrome is X-linked recessive disorder associated with SGPRT's deficiency, the enzyme involved in purine salvage pathway. Okay, the absence of enzyme leads to high level of hypoxanthine. It converts to xanthine and uric acid. This can result in severe gout, renal failure, poor growth, spasticity, and tendency to self mutilation. PCR technique is used for the hybridization of DNA, cloning of the DNA. Yeah, recommendation of the PCR technique basically used for the hybrid cloning of the DNA. Basically, it increases number of DNA. Okay, so basically increase the number of DNA. So this basically this technique is basically developed by 𝘾𝙀𝙎𝙎𝙎𝙬. Okay, 1983, and in 1993 he got a Nobel Prize for this. So this basically three steps: uh, denaturation, annealing, and amplification or synthesis. Okay, first stage the denaturation is takes place. Now the primer will bind. After primer bind, the whole synthesis will takes place. Okay, Okazaki fragments are formed during synthesis of leading strand of DNA, lagging strand, base section repair, or direct DNA repair. So this is basically occurs due to synthesis of lagging strand. During synthesis of lagging strand, they are basically synthesized. Okay. So last question of the today's session, which of the following is true about structure of nucleosome? The right answer is the nucleosome is a unit consists of histone core and linker DNA. Okay. The package of unit of chromatin is called as nucleosome. The nucleosome formed in histone 8 nuclease. Okay. On average, a nucleosome are spaced about 200 base pair apart. Okay. Thank you. Thank you for the joining my class. In the next class, we will come with a new topic. Okay, new topic of discussion. We will meet in the next class. Thank you.